Hello everyone, welcome to the class. So before going to start the class, can you tell me whether I'm audible and screen is visible? Okay, so I will launch the poll. You can tell me whether I'm audible and screen is visible. Okay. Okay, so let us start the class. So before going to start the class, so here is the question for you. Okay, yeah. So in the last class, we have studied about the biotechnological application in the agriculture, right? Okay, so there's a question. See here, the question is, Bt toxin is harmful to the insects, like option A, lepidopterans, and option B is the coleopterans, and option C is diphtheran, and option D is all of the above. You can answer me through the poll, and you can answer me in the chat box also. I will run the poll. Yeah, you can answer me. Okay, fine. I'm going to end the poll. Okay, so here is the answer. So yes, everyone chosen correct answer. The option D. Yes, option D is the correct answer. In the last class also we have studied that, right? So it is Bt toxin will be harmful for the lepidopterans, coleopterans, and also the diphterans, right? So option E, all of the option D is the correct, all of the above. Okay. So in the last class, we have studied about the what is the biotechnological application in the medicine, right? 1 minute. Okay, so just I will recall what are the things we have studied in the application of the biotechnology in the medicine. Okay, so in the last class I have told you, so why this insulin is important. Okay, why we are going to produce this insulin in the biotechnological process, right? So see here, I have told you that this insulin is important because this will take the glucose in our blood and it is going to convert it into the glycogen, right? So that is the reason we require the insulin. So the thing why we started producing the insulin is, the first thing is, it is going to cause the diabetes, right? The diabetic patient could not able to produce the insulin, right? So that is the reason insulin deficiency. They require the insulin, right? So this is the reason we synthesize the insulin in the biotechnological process. See here, in the last class, we also learned that, so what is the, other than the insulin production, so we have taken from the animals also, where it is causes the some allergic reaction. So due to that, we have synthesized it in the biotechnology, right? Okay. Onwards. These are all the things we have studied, and also we have studied the structure of the insulin. What is the structure of the insulin also, right? So this is the mature insulin. So mature insulin, I have told you that it is consisting of the 2 chains, right? Which are those chains? One is the A chain and another one is the B chain, right? So here you can see A chain is made up of the 21 amino acid, whereas the B chain is consisting of the 30 amino acid, right? This is the A chain and this is the B chain. So these all the things we have studied in the last class. Just I am recalling this all the things. Okay. Yeah. So then what is there in between the A chain and the B chain? So there will be a disulfide bond, right? Disulfide bond is there between the A chain and the B chain. Okay. And there is an disulfide bond in the A chain. Okay. So between the chain is there. So that we will call it as the interdisulfide bond. Interdisulfide bond is between the A and B, and the intra means within. Interdisulfide bond is in the A chain. Okay, so this is the thing we have studied in the last class. So then later we have thought that, so there is a one company, so that is started synthesizing the insulin, right? So that can anyone tell me what is that company we have studied in the last class? Yes, so that is the Eli Lilly, right? So Eli Lilly, Eli Lilly company started to producing the insulin. So how they have started producing, I have told you. So it is very difficult to produce the this insulin protein in the bacteria, right? So I have told you that gene processing is different in the bacteria as compared to the eukaryotes, right? So because there is the introns are there in the eukaryotes, whereas the introns are absent in the prokaryote. So that's why we can't able to prepare mature insulin in the bacteria only. So that is why, so what they have did is, so they have taken the gene which is required for the A chain and required for the B chain, and they have synthesized separately the proteins, and then they have added the disulfide bond separately, right? So they have added the disulfide bond separately after purifying this A chain and the B chain. So this is how they have synthesized in this. This is how they have synthesized the insulin in the Eli Lilly company. Okay. So what they have did? First, they have taken the gene separately for A chain and B chain. For A chain and B chain. Okay. So then they have inserted into the plasmid, right? They have inserted to plasmid. What will happen after inserted into the plasmid means? So it is created the recombinant DNA, right? They have prepared the recombinant DNA. See here, this B chain also same thing. Inserted into the plasmid. Then it is created the recombinant DNA. Recombinant DNA is produced, and this thing we have added into the E. coli bacteria. So we know that what happened in the E. coli bacteria. So this in the E. coli bacteria, the plasmid replication is going to be taken place. Plasmid replication is going to be takes place. Then it is going to give more copies of the plasmid. More copies of the plasmid means it is along with the plasmid copies, it is going to give a copies of our related. So that is the A sequence or the B sequence also. So then it is going to convert it into the protein in the bacteria, and then we are going to remove that. Then we are going to recover that protein, recover that peptide. So that is the A peptide and the B peptide from the bacteria. Okay. So this gene, so then it is going to be converted into the protein. You know that process, right? DNA where mRNA is going to be synthesized from the sequence present in the DNA. Then from mRNA, the protein is going to be synthesized, right? So this protein was synthesized in the E. coli. Then we are going to purify this. Okay. So after the purification, what? We have got the, see here, after the purification, we have got the A peptide and the B peptide. Okay, we have got the A peptide and the B peptide, and we take the together that A peptide and the B peptide, and we have added the disulfide bond. Okay, so this is how they have created the insulin in the Eli Lilly company. Okay, and now also we will use the procedure this only. So see here, this is the mature insulin they are given. Mature insulin, right? Mature insulin, where is the A chain and this is the B chain. And here you can see here. So when I have told you in the last class also, whenever the insulin was synthesized, it will be synthesized in the form of proinsulin, right? It will be synthesized in the form of the proinsulin, right? What do you mean by pro? Pro means inactive form. Okay, it is not an active form. So pro means inactive form of the insulin. Okay. So what happened? This inactive form of the insulin consisting of the 3 chains. What is this chain? The 3 chains. One is the A chain, second is the B chain, and third one is the C chain. Okay. So now what happened? So in the synthesis, wherever in the next procedure was went, see here, in the mature insulin, so that C peptide is going to be removed. Okay. See here, the C peptide is going to be removed. only now mature insulin consisting of the only the A peptide and the B peptide. Here you can observe. So there is a disulfide bond and it is the single. So that is the intra-disulfide bond in the A chain. Okay. Yeah. Can anyone tell me which is the amino acid responsible for this disulfide bond formation? I think you have studied in the biomolecules in the first PU. So there are two amino acid. One we will call it as a methionine. Okay. One we will call it as a methionine and second one we will call it as a cysteine. Okay. These two amino acid which is containing the sulfur containing amino acid. Sulfur containing amino acid. Right? Sulfur containing amino acid. But the disulfide bond will not form in the methionine. It will form only in the cysteine. The reason is, you can see here, when it comes to the methionine, the sulfur is there. Other than that, methyl group is also there. Okay. So that is a reason. But when it comes to the cysteine, only the SH group is there. Okay. So that's why. So the disulfide bond will be happen only in the cysteine, not in the methionine. Okay. You have to remember this. Okay. Disulfide bond. Okay. Okay. So if you have any confusion here, you can ask me. Otherwise, we'll go to the next. Okay. Yeah. Okay. So then we'll come to the another very important application of the biotechnology. So that is the gene therapy. So what is this gene therapy? We know what is the therapy, right? So what is this gene therapy? Okay. So we know that what is the therapy, right? Something faulty is there. We are going to replace that by the pure or the some functioning enzyme, functioning things, right? So here what we are going to do in the gene therapy is, so you know what is the congenital disorder, right? What happened in the congenital disorder? Some DNA is going to be damaged or it is going to be non-functional. So that enzyme is non-functional, right? So that in the gene therapy, what we will do, that non-functional enzymes we will remove and we will replace it by the functional enzyme. Okay, functional gene. So that is what we will call it as a gene therapy. Okay, so let us study about this. What is the gene therapy I have told you now? Gene therapy is the replacement of, okay, replacement of faulty gene, okay. Replacement of faulty or non-functional gene, non-functional gene with functional gene, okay, with the functional gene is called as a gene therapy. Okay, so see here, we are going to replace the faulty gene by the functional gene. For example, if some person is having some enzyme deficiency. Okay, for example, if you take some enzyme, okay, so this is the A enzyme. This A enzyme is very important for the, for example, you can take, yeah, I will tell you the example here. Okay, adenosine deficiency, I will tell you. So let us study this. Okay, first. So it is a method for correct gene defect in the child. Okay, 1 minute. Okay, 1 minute. Okay, so it is a method to correct the gene defect in a child or embryo. Okay, in a child or embryo, we can do this. And so here the genes are inserted into the person's cell and tissue to treat the hereditary diseases. It is compensate for the non-functional gene. I have told you, right? This is a compensate for the non-functional gene where we are inserting the functional gene here. Okay, so here you can see there are two types of the gene therapy is there. Okay, so first thing we call it as a, so two types of the gene therapy we can see. One is the germline gene therapy. Okay, first one is the germline gene therapy. Germline gene therapy. And second one is the somatic gene therapy. Somatic gene therapy. One is the germline gene therapy and second one is the somatic gene therapy. So now in the biotechnology, we are doing the somatic gene therapy. Okay. But for till now, they are not approved for the germline gene therapy. Okay. So somatic gene therapy means we know that so all the cells in our body except the reproductional cells, we have the somatic cells, right? So these cells can be called as a if we did any replacement of the gene here means so that we will call it as a somatic gene therapy. Okay, skin cells you can take or any organ cells you can take. So that all are comes under the somatic gene therapy. Okay. So when it comes to the germline gene therapy, if you take egg or sperm, okay, if you take egg or sperm, so if you see here, egg or sperm, okay, 1 minute, egg or sperm. So in the egg or sperm, if you do the gene therapy or if you replace any non-functional gene to the functional gene means that we will call it as the germline gene therapy. Okay. So that we will call it as a germline gene therapy. So for the germline gene therapy, so still the approval was not given. So we are going to go do the only somatic gene therapy. Okay. So I think you understood this. If you are not understood means you can ask me. Okay. So here you can see one of the girl. So that is Ashanti D. Silva. Okay. So this girl is, so in the first gene therapy, okay, this is the first gene therapy. First gene therapy was done to this girl. Okay, the girl name is Ashanti D. Silva in the year 1990. Okay, that too when she was in the 4 years. Okay, 4 years old. Ashanti D. Silva. Okay, so that is in the year 1990. So on the time this girl is the 4 years old. 4 years old. Okay, why we have did this gene therapy means? So this girl is having the deficiency. So that we'll call it as a disease, call it as the SCID. SCID. I think you have learned in the, I think you have heard this name in the immunology chapter. So that is in the health and disease. What is this SCID means? Wait, I will tell you. Okay. Severe combined immunodeficiency. Okay. Severe combined immunodeficiency. Okay. Severe combined immunodeficiency, we'll call it as the SCID. So why these deficiencies happen or what is this? Okay. So what is the reason for this? See here, the reason for this is due to the mutation or loss, okay, non-functional, due to the mutation or you can take the non-functional ADA enzyme, okay, non-functional ADA enzyme. I will tell you what do you mean by ADA, okay, or if you anyone know means you can tell me. ADA means adenosine deaminase, adenosine deaminase. Okay, adenosine deaminase. So I think you have learned in the health and diseases, right? We have a soldier system right in our body. So that is the immune cells we have, right? So what are the function of the immune cells? To fight against the microbes, right? So foreign pathogen inside our body. So for the growth and for the development or the synthesis of that immune cell, this adenosine deaminase is very, very important. Okay. So whenever this adenosine deaminase is not there, the immune cells will not going to be synthesized. What happened then? The person is going to be suffer from the severe combined immunodeficiency. It means it can be susceptible to the infection by the microorganisms, right? So this is the main reason. Okay, so that's why they are given that severe combined immunodeficiency. So what they have did in this adenosine deaminase, what we can do in this type of the diseases, either we can have to do the bone marrow transplantation. There are the two methods to relieve this. First one is the bone marrow transplantation. Okay, bone marrow transplantation we can do. Why we will do this bone marrow transplantation means here the stem cells will be there. Okay, we will call it as the stem cells. Okay, the stem cells will be totipotent. Okay. So this can give rise to the cells. And another thing, another second thing is the enzyme replacement. Enzyme replacement. So by injecting the enzyme, that is the adenosine deaminase. Okay. But these two processes is not the permanent cure for this disease. Okay. This two process is not the permanent cure. Okay. Permanent cure. Permanent cure. Okay. So because these both the things are involved in the somatic, right? Somatic gene therapy. So that is the reason it is not going to be cure permanently. And also so what we have what they have did in the gene therapy means they have taken the bone marrow. Okay. From the bone marrow, they have taken the leukocyte. Okay. Leukocyte they have taken. Leukocyte. What do you mean by leukocyte? That is the white blood cells, right? They have taken the leukocyte. And in the leukocyte, they have removed the non-functional gene. Here you can see. Okay, I will give in the some other color. Okay, wait. Yeah. Let us see. So this is the gene they have taken. Okay. So it is carrying the faulty gene for the adenosine deaminase. Okay. Next, what they have did. So they have removed this faulty gene. They have removed this faulty gene and they have inserted here functional gene. Okay. They have inserted the functional gene here. You can see here functional gene here. And they have grown that in the laboratory. Okay. They have grown that in the laboratory. So they have grown that. Okay. So after the growth, so they have inserted back to the bone marrow. Inserted back to bone marrow. Okay. So this is how they will do the gene therapy. So they will take the one cell from the person who is having the deficiency. So then they will remove that faulty gene. Then they are going to add the functional gene in that. Then they are going to transplant that into they are inserted back to the bone marrow. Okay. So this is how they have did in the case of the Ashanti De Silva. Okay. Okay. So here you have any questions means you can ask me. Here you have any questions means you can ask me. Shaurya Tripathi, do you have any question here? Shaurya Tripathi, do you have any question here? Okay. So let us go to the next, if you understood this means. Okay. Okay. So then we will learn about the application in the medicine. What is the application of the biotechnology in the medicine? So mainly molecular diagnosis. Okay. So here you can see conventional method. So that is the serum and the urine analysis. For example, you can see. So whenever we go to the doctor, so the doctor will treat us based on our symptoms, right? So whenever the person is having any symptoms, the doctor will suggest us to go for the diagnosis. So that is a urine test or the blood test, right? So if we don't have any symptoms, we have to do the diagnosis, right? So for example, if you take the example of the HIV/AIDS, or you can take a simple example that is the coronavirus. The person is not showing any symptoms, so then also we can test, right? So if it is if that virus is in the very small amount also, it can show the positive result. If it is very less amount also, it will give the positive result in the diagnosis, right? So this is what we have to do. This is why it is helpful in the diagnosis. If it is that is in the without the symptoms also, before that only we are going to diagnose it, which disease is that. Okay. So we know that HIV patients also will show the symptoms after some between the 5 to 10 years, right? So before that only we can check that by the diagnosis that whether they are having the virus infection or not, right? So it is very useful to treat the patient, right? So here in the molecular diagnosis, you can see here. So there are so many diagnostic procedures are there. Have you learned about what is the Sutherland blotting? Sutherland blotting. For what we will do the Sutherland blotting? Sutherland blotting. and northern blotting. Northern blotting. And third one is the western blotting. Western blotting. Have you learned these all the things? I think southern blotting you have learned in the fingerprinting, DNA fingerprinting, right? In the DNA fingerprinting, we will do the lastly that southern blotting procedure, right? Yes. So see here, this thing you have to remember. Southern blotting we will do for the DNA, and northern blotting we will do it for the RNA, and western blotting we will do for the proteins. Okay. Yeah. Okay. Okay, ma'am. I can understand. Okay. So hi Jane Vishimla, how are you all? Okay. So let us start. I think you know what is the southern blotting, northern blotting, western blotting. Okay. So can you tell me which is the diagnosis process for AIDS? Diagnosis process for the AIDS? Just I'm thinking, just I want answer from you, whether you know or not. Interact with me. This is the diagnosis process. Have you heard of the ELISA test, students? Have you heard of the ELISA? ELISA. ELISA full form is enzyme linked immunosorbent assay. Immunosorbent assay. Sorbent assay. Okay. So in the exam, they may ask you the full form of the ELISA. Okay. So that's why you have to remember this enzyme linked immunosorbent assay. Okay. So student, it is a test we will do for the HIV AIDS. Okay. HIV AIDS. And for the HIV AIDS, there will be the another test also. So that is the PCR. Okay. Another test also. So that is the western blotting. Another test also. So that is the western blotting. Okay. So these all the things we will do for the ELISA. Okay. So where are these process will come from? So this all the process is the application of the medicine in the biotechnology. Okay. So see here. So it is possible by the techniques recombinant DNA technology. So that is the PCR and the ELISA. Okay. What we will do in the PCR? You know that, right? So whenever we are having the some corona recent days, it is compulsory to take the corona test, right? Means if you are going we are traveling anywhere, or if you want to check whether our fever is either corona or some other, we have to check it by the PCR, right? So that PCR is the RT-PCR, right? So RT-PCR, right? What do you mean by RT-PCR? Reverse transcriptase. Reverse transcriptase. I think you know this, right? Reverse transcriptase polymerase chain reaction. Polymerase chain reaction, right? Chain reaction, right? So this is also the one of the main important things of the PCR. And I think you have learned about the rapid antigen test also, right? Rapid antigen test. Rapid antigen test. Right? What they will do? They will immediately check whether they will take the antigen here. Okay. They will keep the antigen. They will take the antibodies here first. Okay. Antibodies here. So this will give the positive result. See here. This will give the this is for the reference. Okay. This is for the reference. You can check. Here is the Antibodies and antigen both will be there. And here they will keep the antibody. Okay, antibody they will keep. So whenever they take your test, they will keep, they will put here. See here, now what will happen? So this antigen will come and bind to this. Okay. So whenever the antigen and antibody reaction will take place, it is going to give a positive result. Right. So this is for the reference. We can see, have you seen that this will be like this, right? So one is the, whether we have added the positive or negative result, it will show the reference. Okay. So this reference will be the positive only, right? The reference line. So if the person is positive means it will give the line. If it is the person is negative means it will not give any line, right? So this is what we will do in the rapid antigen test. Okay. So you can take the example of the in the pregnancy kit. So these all the things, right? So these all the things, and also the very important thing of the application is in the cancer diagnosis also. Okay. So in the cancer diagnosis also, if we know early that the person is having the cancer means, so we can try to give a treatment and we can save him, right? So otherwise it will goes to the metastasis stage. You know what is the metastasis stage? So that will give rise to the malignant tumor, right? On the time we can't save the patient, right? So this is what the applications of the biotechnology. So that is in the diagnosis process. Diagnosis we will do before the symptoms, okay? Not after the symptoms. Okay, do you have any questions here? Janvi, Shimla? Do you have any questions here in the diagnosis? Okay. So let us go to the next. ELISA, I have told you, right? See here. This is ELISA. What they will do in the ELISA? See here. So first they will put the antibody. Okay. So this is, see here. They have kept the antibody here. Antibody is Y in shape, right? So here you can see this is the paratope, right? This is, we call it as a paratope where the antigen will come and bind here. First they will keep the antibody. Then they will add your sample. Okay. Now what will happen? The antigen present in our sample. Okay. We have, for example, the coronavirus is there, right? This antigen will come and bind to this antibody. Okay. So next what they will do? So another antibody they are going to add here. Okay. Another antibody they are going to add. This is having the some substrate in it. Okay. Some substrate in it. Okay. See here. Target analyte, which is the antigen. So this is the second antibody they are going to add here. Okay. So see here, this second antibody is connected to some enzyme. Okay. See here, this is connected to some enzyme. So then now they are going to put, see here, first what they have did here, it is having the only antibody. We have added our antigen. So that is the sample, right? Sample was added here. So then second antibody they are going to add here. Our second antibody is not a single, okay? It is having the enzyme also, okay? It is having the enzyme linked to this. So that's why it is called as a enzyme linked immunosorbent assay, where our second antibody is carrying the enzyme. So that is the reason, okay? So now what will happen? For this, we are going to add substrate. Obviously, where is the enzyme? It is going to convert the substrate into the product, right? See here. Here, there's one interesting thing is here, okay? So just listen here. So what happened? First antibody is here. First antibody is here. So this is the second antibody which is carrying the enzyme. So that's why I told you that enzyme linked, right? Second antibody is carrying the enzyme which is linked to the second antibody. So see here, then I am adding the substrate, okay? Substrate that is specific to that enzyme. Okay, specific to that enzyme means what? That enzyme will convert that substrate into the product, right? So that will convert the substrate into the product, right? See here, the enzyme linked to that, it is going to convert it into the substrate into the product, right? Okay. So now this solution is going to give a color. Okay, this solution is going to give a color. Then it is going to, we will tell this is the positive result. Okay, then we can tell that this is a positive result. So just imagine here, this antigen, if it is bind to the antibody first, right? So then only this second antibody is going to bind to this antigen. Okay. If this antigen is not binded, means the secondary antibody will not come and bind. Okay. So that is why it is called as it is very rapid. Okay. It is rapidly we can do. Okay. This is the test we will do for the AIDS also. AIDS and for the some other test also we can do. I think, do you understood this? Student Jainvi Shimla, can you tell me whether you understood this or not? Okay. So this is about the ELISA. I think you understood this. Okay. Let us go to the next. Okay. Transgenic animal. So in the before only in the biotechnological process, we have learned that. So transgenic animals we are going to create, right? See how we are going to create a transgene. I have told you that this is the plasmid. We are going to insert the gene to this, right? So when it comes to the transgenic animals, so we are not going to insert it into the somatic cell. Okay. We are not going to do into the somatic cells. Okay. Somatic cells, not to the somatic cells. We are taking into the, we are inserting our gene of interest to the zygote. Okay. So we are going to insert into the zygote, or we can introduce into the, into the zygote. 1 minute. In the zygote. What do you mean by zygote? What do you mean by zygote? When egg and sperm will combine, when the fertilization was happen, on that time it is going to form the zygote, right? So we are adding the cell. Which is that cell? Recombinant cell. So this is the recombinant DNA is there. This is carrying our inserted gene. We are going to add that into the zygote. Okay, then what will happen? It will insert it genetically. Then it was going to be show the result, right? So that's why we are going to insert into the zygote. Okay, so this is thing we will call it as the transgenic animal. Transgenic animal. Okay, so what is the speciality of this transgenic animal or inserting the gene into the zygote means? So whenever the offspring was produced to this transgenic animal, it is the gene which is inserted, right? That will goes to the offsprings also. Okay, it is going to the offsprings also, to the children, right? Children of the transgenic animals also, right? So that's why we are going to insert into the zygote. What if we insert that into the somatic cell? What it is going to be happen? It will not going to be transmitted to the offerings, right? To the children, then it will be waste, right? So transgenic animal will carry the gene. It will transmit to the next generation also. Okay. Yeah. Okay. So then we learned about, first we will learn about what are the application of this transgenic animal. Okay. So there are animals whose genome has been altered by introduction of an extra foreign gene by the manipulation. Students, here you have to think, okay, whichever the genes in our body, fine. Whichever the genes we will insert from the outside, right? So that all the genes we will call it as a foreign gene or the extra gene. Okay. So whenever the foreign gene comes to your mind, you have to think that this is a gene it is not present in that animal. Okay. Then see here, we have produced so many transgenic rats, rabbit, pigs, sheep, cow, and also the fish. Okay. So over 95% of the transgenic animals are the mice. Okay. So usually we'll produce a transgenic animal. So that is the mice for the medical purpose and for the vaccine purpose. These all the things. Okay. Let us see one by one. You can see here, it is going to show the radium. Okay. See here, they have added the radium gene in this. Okay. Radium. So then it is going to show the color. So here in the rabbit also. So see here the sheep and the pigs and in the cow and in the fish. Okay. Let us study one by one. Okay. So what are the benefit of this transgenic animal? Why we have to produce this transgenic animal? Yes. Without the use, we will not create any transgenic animal in the biotechnology. Okay. Let us study one by one. So to study the regulation of gene or the normal physiology and the development. Okay. I will explain one by one. Okay. Just I will tell you. And second one is the contribution of the gene in the development of the disease and biological products and vaccine safety testing and also the chemical safety testing. Let us study one by one. Okay. So see here, to study in the regulation of the gene in the normal physiology and the development. Okay. For example, if you see, we don't know what is the function of that gene. Okay. So what we will do, we will take the mice. Okay. See here, mice we will take, genetically modified mice we will take, and we will remove some gene. Okay. We will remove some gene, particular gene, not some gene. We will remove particular gene from gene. Okay. Not some gene, particularly gene. Okay, sorry, 1 minute. Okay, see here. So I have taken the mice. Okay, I have taken the mice. In the mice, I have taken the chromosome number 2. Okay, I have taken the chromosome number 2. Okay, chromosome number 2 I will take. Okay, so what I will do, I will select some particular gene in the chromosome and I will delete that. Okay, so I will delete that gene. Okay, I will delete that gene, and that after that deleting that gene or removing that gene, I will observe is there any functional differences is going to be taken place in that animal. Okay, so anything is going to be happen in that mice. Okay, so it is going to show some defect. Okay, it is going to show some defect. Okay, for example, that gene is very important for the heart development, or that gene is very important for the bone development, or that gene is very important for the tail development. Okay, so if I, after removing this gene, so there is no tail formation. Okay, the tail is not formed. Okay, tail is not formed, right? Tail is not formed. Or you can take the brain development was not happened. So on that time, for the studying these purposes, we'll do the transgenic animals. Okay, I think if you understood this, means, can you tell me yes or no? Anup Kumar and Rachna, can you tell me whether you understood or not? To study the physiology and the development, I have told you, right? If I delete that gene, particular gene, I'm not able to see that brain development in that particular mice, or any tail formation in that particular mice. Then I came to know that this gene is very important for the tail formation, or this gene is important for the brain development. Okay, so thereby we can study the importance of the gene, right? Importance of the gene and what is the function of that gene. Yes, yes. So that is what we will do in the studying the normal physiology and the development. Okay. Okay. So then we will come to the study the contribution of the gene in the development. 1 minute. Development of the disease. Right. Okay, Rachna. Okay. So we know that if we do any mutation, okay, I have told you in the same thing also. So there's some enzyme is there. Okay. Some enzyme. For example, if I take the insulin gene, okay. So 1 minute. Okay. For example, I have taken the insulin gene. Okay. In this mice, I have taken the insulin gene and I have mutated that, or I have removed this insulin gene. Okay. Can you tell me which disease is going to be caused to this mice? Can you tell me which disease is going to be caused to this mice? Which disease is going to be caused to this mice? Diabetes, right? Diabetes. Diabetes is going to be caused. Okay. So 1 minute. If I remove this, diabetes, it is going to be caused, right? So this is what I have told you in the study of the physiology. Okay, not for the disease. Physiology. So another thing, so that is in the study of the developmental of the disease. For example, I can take some bacteria. I will take this transgenic mice, and I can inject some virus or some bacteria to this, and I can see what are the symptoms this mice is going to show. Okay. And thereby I can study the disease also, right? If it is infect to the persons or to the humans means, it is very difficult, right? So before that, we can inject that into the mice, either the virus or the bacteria. Thereby I can study what are the symptoms this mice is going to show. Okay. So this is how we will do. Okay. So we will inject, or we can make this transgenic mice to expose to the bacteria or the viruses. Okay, either virus or bacteria or some pathogen. Okay, bacteria. Then it is going to show some symptoms. Okay, it is going to show some symptoms. Thereby, it is easy to study the disease, right? Easy to study about the disease. So this is the one more thing. And let us go to the next. So biological product. So here you can see here, alpha trypsin. Okay. Alpha trypsin is a biological product which is produced inside the mice. Okay. In the mice or in some model organism, in the transgenic animals, we have synthesized this alpha trypsin protein. Okay. This alpha trypsin is nothing but the protein. Okay. So how can we use these transgenic animals? Transgenic animals, we can use it for the production of the biological products. Okay. So this alpha trypsin, we produce it in the transgenic animal. How we have prepared this alpha trypsin? We have inserted the alpha trypsin gene in the mice or any model or any in the transgenic animal. So after that, it is inserted. It is start producing the alpha trypsin. Okay. So then we will collect some samples. Either the blood we will take, or either the some urine sample or some sample we will take, and we will check this protein in the transgenic animal. Okay. For example, I will tell you that we have prepared some protein. Okay, that is milk in the rosy cow. Okay, rosy cow. Okay, in the rosy cow, we have prepared the alpha protein. Okay, this protein is very rich. Okay, this protein is very rich and it is having the 2 gram. Okay, 2 gram per liter. Okay, 2 gram per liter, and it is useful for the newborn babies. Okay, newborn babies. Okay, so directly we can give this milk to the newborn baby. It is having the very high protein, right? So this is the biological product. So biological product, how we are going to synthesize? By using the transgenic animal. In the rosy cow, we have inserted this alpha protein gene in that. Then in the cow milk, it is going to produce the alpha protein. Okay. Then I have told you about the alpha trypsin also. So this is also produced in the transgenic animals, and the alpha trypsin is used for the treatment of emphysema. Treatment of here, it is treatment of the emphysema. What happened in the emphysema? So we are going to reduce the surface area for the exchange of the oxygen and the carbon dioxide. Okay. So in the emphysema, you will study in the respiratory disorder. It will decrease surface area. Okay. Surface area for oxygen and carbon dioxide exchange. Okay. So this is what? So the help of the alpha trypsin protein. Okay. So I think you understood this. So another thing, main important. So we'll study about the vaccine. So in the corona time also, so many of them everyone waited for the vaccine for the corona, right? So till in the news, we have heard that still it was in the first testing, second testing, third testing is going on, right? So whenever the vaccine was prepared, so it was first tested with the cells, then with the transgenic animals. Okay, so here you can see here. So whenever it was first synthesized, it will give rise to the mice. Okay, so then they will observe that. Okay, first they will observe that. Is there anything is going to be happen to that mice? Okay, nothing was happened, and it is started to produce the antibodies. Then it is successful. Okay, so then it is going to be successful. Then we are going to the next step. What is that next step? Then after the mice, we are going to inject into the monkey. Okay, monkey. So then we are going to observe that So after that, anything, any side reactions or any allergic reaction, something is not happened means, then we are going to give for the humans. Okay. So that is the 3 types. So that is the testing we will do in the vaccine testing. So that is in the transgenic mice. First one is the transgenic mice we are going to do. And secondly, we are going for the monkey. And thirdly, we are going to do for the humans. Okay. So when it was come to know that it is a safe, then only we'll give to the humans. Okay. So this is about the vaccine testing. Okay. So then we'll go to the toxicity testing. So you know that some chemicals will cause some disease, right? So we are the humans. We don't have any we are strong. So we don't have any of the reactions means if we give this any toxicity means, for example, you can see here the normal mice. Okay. You can take the normal mice. The normal mice is going to react to the chemical very fastly. So you can see here 1 gram of the chemical. Okay. Any chemical you can take. Okay. So 1 gram of the chemical is required to show toxicity. Okay. Toxicity to this mice, normal mice. So what we will do, we will create the transgenic animal. So that is, we will make them weak. Okay. We will make that weak. Okay. We'll make that weak. So that is susceptible to the, it is susceptible to the less toxicity, less toxic chemicals. See here. See here. For example, I have told you that in the normal mice, to show the toxicity, it must require the 1 gram of the chemical, right? So here it is going to show for the 0.001 gram. Okay, 0.001 gram only sufficient to create the toxicity in this transgenic mice. Okay, so that is another important. So that is the chemical safety testing. Okay, so they will make this mice into the weak. Okay, weak. Then we are going to use that for the toxicity testing. Okay. So do you understood this? Do you have any questions here? Okay. So if you don't have any question means we'll go to the ethical issues. Okay. What is this ethical issues? Okay. So whenever we are creating any transgenic animals here, any transgenic organism in the biotechnology means, we should think of our society, right? Whether this is useful to the society or harmful to the society, right? So whether it is useful or harmful to the society, right? So there are some rules will be given. Okay, some rules will be given in the biotech. Rules will be given. So based on that, we have to do for the the genetically modified organism. Okay. For example, if you think any terrorists have modified some organism and they have used it as a bioweapons, so then what will happen? So it will be dangerous, right? So any creating any genetically modified organism, there will be a rules will be given. Okay, rules, and we have to take a permission from the 1 minute. We have to take a approval from the GEAC. 1 minute. GEAC. What do you mean by GEAC? Genetic Engineering Approval Committee. Okay, Genetic Engineering Approval Committee. Okay, so we have to take a permission from the Genetic Engineering Approval Committee. So then only we can use that genetically modified organism. Okay, so otherwise some wrong things going to be happen. So that is the reason. Okay, so one more thing here you have to remember. So whenever, so there will be a patent will be taken, right? Patent. What do you mean by patent? For example, I have did some experiment. Okay, so till for 10 years I have did some experiment. I have created some product. Okay, some product I have created. So now that is I am going to sell that medicine for the some 100 rupees or the 200 rupees. Okay. So in the next, after the 1 or 2 months, some other company started to produce that same product or the same medicine, and they have used it to sold for the some less amount. Okay. So then what happened? So I have taken for 10 years to prepare that. They have taken only for the 1 month or 2 months to prepare. They have taken my ideas and they have started to prepare that. Right. So that is the reason whenever the person created first product, they are going to take a patent. Okay. So patent means, so that is a right. Okay. That is a right given to the person. That is a right given to the person who produced first product. Okay. Person who produced it, or who, yeah, we can take the who produced first product. Okay. So when it comes to the product, we'll call it as the patent. Okay. 1 minute. When it comes to the literature, if you have written something, and so that is written by you first time and it is going to be patent. So that we will not call as a patent. So that we will call it as a copyright. For example, if you have created some procedure for some experiment, or first time you have created something in the, you can take some procedure. Okay. So that we will call it as a copyright. Okay. The writers will take the copyright. Okay. If the product was produced, means they will take the patent. Okay. So if it is in the biological, so that's why we will call it as the bio patent. Bio patent. Okay. So see here, something was inserted, some gene was inserted. So see here, if the humans was born like this, means it is a problem, right? Okay, so that is comes under this thing. So now let us talk about the biopiracy. What do you mean by biopiracy? Okay, for example, you can think, 1 minute. Okay, so let us just continue this biopiracy in the next class. So whenever you are coming to the next class, you learn and come to this class. Okay, nothing. Biopiracy means, so whenever the things was created in the one country, so without the permission or without giving any payment to that company or that country, we are selling in our name. Okay, we are selling that in our name by creating some other name. Okay, so that is what call it as the biopiracy. Okay, for example, here I have taken the example of the basmati rice. Okay, in the basmati rice, you can see here. So basmati rice. Okay, basmati rice is the Indian origin. Okay, but some American company have taken this and they have made a patent. Okay, they have made a patent. Okay, then they have taken a patent of that and should not be sold by the India. So, but after that, it came to know that this is the basmati rice. We are growing that from the so many years. So then it patent was cancelled. Say second thing is the turmeric. Okay, the turmeric is having the antibiotic properties and the herbal properties, right? So that is also some companies try to make a patent of that. So that is also not possible because the patent was not given to the herbal product. Okay, no, patent was not given to herbal product. Herbal product. Okay. And also you can see here neem. Okay. Neem is also having some antibiotic property and some medicinal property. So that is also some company made to make a patent of that, but it is also failed because the herbal product. Okay. So these all the things, these three things are from the India. Okay. But some other countries have taken this advantage and they have made that without giving the Indian name. So they are used to take their name and they are going to take the amount in that name. So that is a reason. So this is what we call it as a biopiracy. Now, no problem. In the next class, we will learn about this biopiracy in detail. Okay. Just I have given the introduction. Fine. So let us continue in the next class. So this is a homework question for you. Okay. So this is a homework question for you in the next class whenever you are coming. So you can solve this and you can come. Okay. Yeah. Thank you so much. We'll end the class now. If you have any questions, you can ask me. Or you can leave the class.